previous homeworks.
So the homework that you have to pass to me is... Okay, thank you.
Thank you. All right, so did you have a good Halloween? I think that's where I ended. And then we had a guest lecture by Jingyi Fei last week, and then you had a lab tour. Okay. So I believe she discussed two single molecule based methods. One is FRET, the other is super resolution imaging, and I have one more trip later this week. So Professor Yan Shemna from physics will give a guest lecture on Thursday. It'll be on optical trap or optical tweezers and its applications to biophysics and cell biological problems. Okay. So today, I actually want to talk about the combination of FRET and force using a work done in my lab as an example, and mainly because you'll be doing a homework problem on a related question. So I just wanted to give you a context. Okay? As you know, DNA is a double helical structure. And in the canonical form, you have two strands of DNA forming a double-stranded DNA. But in some special circumstances in the cell, DNA can also assemble into a four-stranded structure. You can have four strands of DNA assembling into a four-way junction. And this junction structure is actually important for DNA repair. And it's also called the Halliday junction with two L's. That's because more than 50 years ago, Dr. Robin Halliday proposed this as a structure of DNA. Okay? And maybe can you turn down the light? All right. So this is, that's good, yeah. So that is the four-way junction structure. In fact, the molecule takes the structure shown here when you have no ions in solution, no magnesium ions in solution. And this structure is called the open structure. It looks like the four arms of the four-way junction are pointing four corners of a square. And but that will be highly non-physiological. In the cell, you always have cations, a lot of divalent ions, magnesium and calcium and so on. So in fact, when you have ions in solution, then this molecule changes its shape into a structure shown here. That is a crystal structure of the four-way junctions solved by some people some years ago. And it looks like an X shape. Okay? In fact, you can see that one strand follows continuously in this direction and so on.
Why is it 3 colors?
Oh, actually, there are 2 different red colors. So it's not, you know. I think I didn't do a very good job. I believe I made that one. Okay. So the way it actually forms into this structure is shown here. So you have 4 cylinders in 4 different colors, and they can actually stack on top of each other. You know, I discussed stacking between 2 books or base pairs. So you can see that green helix can stack on top of the blue helix. On top of the red helix, forming this. If you want, you can have 2 beer cans stacking on top of each other. Okay, and then blue on top of the white. And then these 2 super helices form a small angle. So this is called a stacked X structure because it looks like an X, and then you have stacking between different helices. And this stacking of helices is actually very commonly seen in various RNA structures found in nature. But as you can also imagine that by symmetry, that green guy can also stack on top of the blue helix, not just red one. Okay, so there is actually a related structure shown here. So in fact, these 2 are 2 possible conformations, or isomers, if you want to call them. And we have shown in my lab that these 2 structures can convert between the 2. Most likely is through this open structure as an intermediate. Okay. And the way we can look at this is by attaching the fluorescent molecules for FRET measurements, green and red, donor and acceptor. And when it goes from this state to the other state, then 2 dyes come close to each other. So when you excite the green molecule, you get strong energy transfer, you get red signal. So that is a really simple readout that you can get. So this is the idea, that this guy can go between one stacked X structure, possibly through the open structure, to the other stacked X structure. And you can immobilize a molecule to a surface using, a well-known molecular glue called biotin-avidin interaction, and they can shine light to follow the motion. Actually, I made this movie many years ago, teaching myself how to use a ray tracing program. But although the structures are known for the 2 extreme states, how actually it goes from one state to the other was actually not known at all. So this is, in a sense, a pure fantasy. I just use some mathematical interpolation to make these intermediate structures. So but if you actually do a single molecule FRET measurement, you measure energy transfer efficiency as a function of time from a single 4-way junction. And what I'm plotting here is energy transfer efficiency. So when 2 are close to each other, in this form, state A, then you get high energy transfer. So you get high value of efficiency. And in the other form, state B, you get low value of efficiency. And then you can see the molecule going back and forth between the 2 states. And you can also plot the histogram of the 2. And then you see nice two peaks. So that was actually one of the first papers we published from my lab in Illinois. you can also do this as a function of ion concentration, so varying the magnesium ion concentration as you decrease the ion concentration from 50 millimolar to 3 millimolar. You can see that same two-state fluctuations still occur, but much more rapidly. You can even measure the rate of transition rates for the forward and backward reactions. Both of them actually goes down as you increase the magnesium concentration. As you increase the concentration, everything slows down. Rate is going down in both directions. Okay? And the rationalization for this was the following: that to go from one state to the other, you have to go to this intermediate state. Therefore, you have to unstack all the stacking interactions. But here, magnesium ions actually bind tightly to the junction because there are a lot of negative charges there. So if you have more ions in solution, then each of these two structures becomes more stable. So it becomes more difficult to leave to go to the open structure. That's why the reaction is slow and slow as you increase the ion concentration. So you can, and that's why when you change the ions, everything goes down. The next thing that we did was to, use it as a model system to develop a tool to apply force to a single molecule and then measure how the molecular conformations and the dynamics are affected by the applied tension. So how much force is needed to make some changes, and can you learn something about this, especially what we discussed 2 weeks ago, transition state. Can you deduce some information about the structure of the transition state? Okay, what is the intermediate pathway for the molecule to switch from one state to the other? So the idea is the following. So now you have two states that are more or less equally populated. Then what happens if you use optical trap that you hear on Thursday to apply force to one arm of the DNA? You see that you have two arms with dye labels, and then you have one arm for surface attachment. You have one free arm. Let's apply force to this one. Okay? And then which do you think will be actually preferred if you apply force? A or B? B. Why? Because the distance between the two force application points is larger there, right? So that state will be preferred. But of course, before you measure it, you don't know how much force it will take, and so on. So that's what you expect to see. And one rate will go up, the other rate will go down, and so on. Right? So we did that by extending that arm by using a much longer DNA, about 16 microns long, 50 kilobase long lambda phase DNA that I discussed, and we can attach the other end to a small bead that you can grab using a focused IR laser light. Then you can apply force of a defined amplitude and measure Fred using a focused laser light. And here we are using two laser beams that are spatially separated, so you don't have to worry about interference between the two laser beams and what they're trying to achieve. So here is the Fred time trace at a very low force, 0.5 piconewton, and you can see that you see two-state transitions that are more or less equally populated. And then now we increase the force. Well, the effect is subtle, so you go to higher force, and you start to see that you see more and more in the low Fred state, and then the effect is now pretty obvious. And then as you go to even higher force, then now it's mostly in the low Fred state with brief excursions to high Fred state. And this actually matches the expectation because of the way we are applying the force. We expect to see this state to be favored, but this one should have a larger separation between the diamolecules and low Fred efficiency. So that is actually exactly what we are hoping to see. And so when my postdoc obtained the first set of data in the middle of the night, he was so happy that he got a speeding ticket on the way back home. And you know how difficult it is to get a speeding ticket in this town anyway. So then you can analyze the data using hidden mark modeling to obtain the rates in an unbiased way. Then plot the rate of transitions between the two states as a function of force. So that is shown here. So force and the log of the rate here. And so green is for the forward reaction and red is for the backward reaction. And as you can see that as you increase the force, the rate of forward reaction increases. The rate of backward reaction decreases, and as you expect. But what we didn't expect to see was that the slope was not the same in both directions. So in the forward reaction, slope was smaller than the backward reaction. And what actually it means is that if you think of these two AB states as energy minima in this free energy diagram, that transition state that we discussed must be closer to the state A than state B. Why? Because if you apply force, you are tilting the energy landscape by adding a linear potential in this direction. So you are changing things this way. But because of small separation, the change in the barrier height for the forward reaction is not as big as the change in the barrier height against the backward direction, meaning that the force will have a smaller effect in this direction compared to the backward direction. And that's exactly what you see. Okay? So just based on the slope, this is about a third of the way to the other direction. So that is the prediction. But what actually breaks the symmetry to give you this type of asymmetry? So my argument was that, well, if you actually assume that there is a stable intermediate as the open state that I discussed, then you'll have to have two high energy barriers to cross, and that are more or less equally populated. But as soon as you apply force to tilt the energy landscape, then you pick the winner out of the two. So this becomes a major barrier. It is close to the state A on the left side. That's why that's why you see the difference in slope between the two reaction rates as a function of force. The prediction of this model is that if you then tilt the energy in the opposite direction by applying force in a different direction, then that should become the major barrier, and then you'll get slopes that are changed accordingly. So if I show you the experiment, so this is the original way of doing the experiment, but you can actually use just the DNA manipulation to pull on different arms, like that, and then we can measure the same thing. And it turns out in this case now, the forward reaction shown in green has a higher slope than the backward reaction as predicted by our model. So that was nice. And then finally, we applied force in the remaining possible way, because initially we applied force this way, and then this way, and then we can also apply force in this direction. Then ask, what's going to happen here, and we did see a force dependence, but not very strong. But in this case, both of the rates actually increase as you increase the force. So based on the data, determining the transition distance to the distance to the transition state in all three pooling configurations, we are able to propose that in the transition state here and here, what you have is a structure that is really like an open structure, except that instead of having a square in the middle, you have a diamond in two different directions. And so that was actually a paper we published in 2007, and you'll be doing a homework problem, basically we plotting the data and then use what you learned trying to calculate the distance to the transition state and answer some other questions. All right, any question about this part? Okay, let me move on. So we are done with a single molecule biophysics portion of the lectures, and we'll be moving on to more systems biology type of materials. So again, you can turn on the light. So let's just remind ourselves what we are talking about. So here are two black lines. I'm denoting the DNA, okay? And then this much of the DNA is the gene, encoding the sequence necessary to make RNA and then protein. And here, this segment of the DNA is called the promoter. What it means is that it has a special sequence to which the RNA polymerase can come and bind and start transcription of a new RNA molecule. So normally, RNA polymerase is able to come and bind if you present naked DNA to the molecule. But as you discussed, in the cell or in the living organism, it's important to regulate gene expression because you don't want to make, proteins all the time. You want to make them when you need to, and exactly the amount that you need. So, so what usually happens is that there is actually a special sequence that we can call operator, and to which another protein can come and bind. And that protein, in this case, can be called the repressor protein. So when the repressor protein binds to that DNA segment, then promoter is actually blocked so that RNA polymerase cannot come and start transcription. So you can repress gene expression by allowing repressor protein to bind to the segment. So I gave you an example of lock repressor that E. coli actually makes. Usually lock repressor binds to this location to prevent the transcription of genes that are required to process lactose sugar. But when lactose level goes up and then glucose level goes down, then it'll dissociate to allow RNA polymerase to bind to the promoter to start transcription. So I think glucose and lactose is like ice cream and broccoli. So if you give the E. coli if you give my children ice cream and broccoli, they will eat only the ice cream. Right? And only when they run out of ice cream and nothing else to eat, they get hungry, then they eat the broccoli. Same here. So E. coli will, first use glucose, but when there's no glucose and lots of lactose, then it'll actually make lactose utilizing genes by making the lock repressor dissociate from this operator, and so on. So this was a quick review. Any question? So here is a cartoon that I took from a page of a textbook by Rob Phillips. Here in real E. coli cell, you have the promoter site, and then from which transcription can begin. So this arrow, little arrow that I show here is marking the transcription start site, and the arrow points in the direction of transcription. So gene is downstream here. So that's where the promoter is. And then white rectangle denotes the operator site to which the lock repressor can bind to repress transcription. So when the lock repressor is not bound to the DNA, then this gene can be produced as a protein. And if you fuse that gene to the gene of a DNA sequence encoding green fluorescence protein, then fluorescence protein can be produced, and the E. coli cell can light up. Okay? So you can use that fluorescence signal from the cell as a signal for transcription of the gene. But if you have a lock repressor binding to that operator, then it will prevent transcription, and the gene it becomes repressed, and the readout will be a much lower fluorescence signal of the GFP produced. One interesting complication here is that there isn't just a single operator sequence that binds a lock repressor. There are actually two of them here, minimum. And the maximum repression can be achieved only when the lock repressor binds both of them simultaneously, forming a DNA loop between these two white patches bound by the same protein complex. So that's when you get the maximum repression. Okay? Now, people have actually done really a crazy experiment. They can actually engineer the DNA in E. coli, so you can change the distance between the two white patches for operator distances between many different values, down from 55 base pairs to 155 base pairs. And then they can measure the level of repression. Okay, when this is high, it means that this type of loop can form easily, so that you can repress the gene expression very nicely compared to the unrepressed state. So you can repress that by several thousand folds. Quite impressive. Now, as you can see, you see actually peaks, nice peaks, spaced by about 10 base pairs. That actually comes from the helical structure of the DNA. Okay? When the protein complex binds two sides of the DNA, but protein cannot really distort itself too much. But the DNA has to present itself in registry with the protein structure. If that is not matched well, then it doesn't bind that well, so repression level drops. Okay? Only when that registry is perfect, you see these nice peaks here. So that is not actually very surprising that you see this 10 base pair periodicity. The surprising thing about this data was that the peak of repression occurred somewhere around 70 base pairs. Why is it surprising? Because for double-stranded DNA, the persistence length of the DNA is about 150 base pairs, or 50 nanometers, so around here. Meaning that once you go below this length scale, it's really unfavorable to actually form a DNA circle like that, because DNA is not really flexible according to that classical model at this length scale. So if anything, your expectation should have been that the envelope function, instead of showing a peak here, it should show a peak somewhere up here. So that was a surprising finding, and in fact, how flexible DNA is at this length scale is actually a highly controversial topic. Okay? Yes.
So when they were inserting sequences to lengthen the distance,
yeah,
were they inserting random sequences?
Actually, I don't exactly know what sequences they inserted, and but that is a really good question. And Rod Phillips actually has repeated this experiment, but now for the same distance, but took two different sequences. and with which they can also measure the flexibility of the DNA. But in vivo, there was absolutely no sequence dependence of the intervening DNA sequence, according to their experiment. But well, one thing I have to mention is that usually people talk about in vitro artifacts. In vivo is real and in vitro can be artifactual. But here is an example where you have to also worry about in vivo artifact. Because if you're interested in how flexible the DNA is in a naked form, then in the cell you have many other proteins that can bind to the DNA that can complicate your interpretation. Questions? So the key point that you need to remember going forward is that you can have on-state and off-state. So when there's no protein bound to it, RNA polymerase can bind and then make proteins at a steady rate. And here, the rate will be very low, if not zero. This is the E. coli genome, and about 5 megabases long. And this is where replication starts. It's called original replication. And log operand actually is sitting somewhere here. And somewhere here, there is a binding transcription start site. Transcription occurring in this direction. And then you have operator sequences and here and then downstream, so you can log repressor can bind and form a loop. And once transcription starts, then you make beta-galactosidase that can break down the lactose sugar, and then lactose permease that can import more lactose molecules from outside. And then this is an enzyme also that processes a downstream product. So you can actually make them all in one shot, to utilize the sugar available. But this actually log repressor and then this promoter has been used as actually a very useful biotechnological tool to make genetic switches and circuits and so on. So I describe one application here. So it's called the toggle switch. So there, and just like the light switch in the room we have, it has two stages, right? So it's really hard to maintain the switch in the middle position without continuous feedback, right? And in fact, it has no defined state there. So you want it to operate as on or off switch, okay? And so what is the minimal requirement to make such a toggle switch in gene expression? And that is shown here. So one is gene for protein 1, and 2 is the gene for protein 2. And then when the protein 2 is produced, then this can go and bind to the promoter of region of the gene 1 and then represses. So this flat line here means that gene 2 will actually repress gene 1, and gene 1 will also repress gene 2. And that can give rise to a toggle switch for the following reasons. So if you have circuit like this, and for one reason, let's assume that the level of protein 1 goes up, right? then this will become more efficient in repressing the production of protein 2. Right? So protein 2 level will go down. And because protein level of the 2 goes down, then it will be less efficient in repressing protein 1. So then 1 will continue to be up, and so on. So actually, what end result is that you get this, that, and then it just stays like that. Right? But if due to some other fluctuations, protein level actually goes down, then you go to the other state of the switch. So that is like minimum functional toggle switch you can imagine building. The way you build it is illustrated here. A little bit complicated. So repressor 1, the protein 1 is made, and then it can bind to the promoter sequence that it can recognize to repress the production of repressor 2, protein 2. And when the protein 2 is made, then it can also bind to its recognition sequence here to repress the expression of protein 1. So they actually repress each other. And you can read out the outcome by fusing one of the 2 proteins to a GFP as a reporter. Okay. And here I'm denoting inducer 1. And so that is, if you want to switch from one state to the other, you can go and apply force to your light switch. Right? You can actually force the system from one state to the other by adding an inducer. And then this will result in actually a dissociation of the repressor 1 from that promoter to allow the repressor 2 protein actually go up in expression. So you can actually change the state of the switch using an external inducer, just like you can change the state of the switch by using a mechanical force. So this was done about 13 years ago by the lab of Jim Collins at Boston University. So this is really a famous Nature paper, and the first of really its kind. And what they do is that they change the inducer concentration, which is a sugar analog, for lock repressor. So as you increase the lock repressor concentration, as you increase that inducer concentration, then eventually lock repressor dissociates from the DNA, and then you are flipping the switch to the other state. And when you look at the cell population, then you, instead of seeing a gradual fluorescence increase, you see a sharp increase right at a defined concentration, suggesting that yes, indeed, you are looking at a switch-like behavior. You can also measure the fluorescence of single cells because of the GFP reporter. And on the one condition, shown in condition 2, somewhere here, where you see cells have a low fluorescence. I'm sorry that the labeling is actually done in opposite orientation. So as you go to the left, fluorescence signal goes up in the log scale. But as you increase the inducer concentration, you go from here to there, and you can see that guy, low fluorescence cells, disappear as a population, and then you see the emergence of the high fluorescence cells. Okay, so when you dial up this inducer concentration, you don't see cells shifting gradually from one state to the other, but one state disappears and the other state appears, function like a toggle switch. So the paper was very important. It's still very important because if you put this type of circuit into the cell, there's no guarantee that it'll actually work, because there are many, many cellular proteins also there. And how do you actually make sure that your circuit does not perturb the cell, or cell does not perturb your circuit? So when it actually works, it's a cause for celebration. Question?
So what was the IPTG doing? Can you go back on that?
So I think here, I think you can use this. So you can then add IPTG. Okay. Then when IPTG is added, then you can repress the repression. Okay. Then this repressor 1 protein will now be produced and giving you GFP signal. Okay. But if you don't design a toggle switch, generally what you expect to see is that as you increase the inducer concentration, you'll simply see cells getting brighter and brighter. Instead of just showing two off and on state, you see all these gradations. But that's not what they say. They see a switch-like, behavior where you see only off or on. And here is another example data. So you can actually add the inducer at a certain concentration and measure the cell fluorescence signals from single cells as a function of time. Okay. So initially there's only 3-hour time points. Then now the labeling is up, the reverse. So the fluorescence is weak for older cells at 3-hour time points. But as time goes on, at 6 hours, all of the cells switch to this high fluorescence state, on state. But in between, you see them actually toggling from off to on state, instead of just gradually shifting in position. Okay. So that is the idea behind the toggle switch. Okay, so I come to the math part after we actually discuss the other two examples. One is called Repriscillator, because it's an oscillator made of repressors. So this is a paper published also in the same month in Nature by a group of Stan Leibler at Princeton. And what they wanted to do was to build actually a circuit that oscillates. Instead of having two states, you want to have a circuit that goes up and down in amplitude as a function of time. Just like a circadian rhythm, some protein levels can oscillate with a 24-hour cycle in our bodies. And they wanted to actually build an artificial circuit that oscillates in time. And that is what they did. And briefly, you can view it in this manner. Now you have 3 different proteins, 1, 2, and 3. And 1 is repressing 2, and 2 is repressing 3, and 3 is repressing 1. Okay. And how do you get an oscillation out of this circuit? Let's imagine that protein 1's level goes up. Okay. Then this guy will repress protein 2 production. Therefore, protein 2 level will go down. And then it'll be less efficient in repressing protein 3's production. Therefore, this guy will go up, and then it'll be efficient in repressing protein 1's production. So protein 1's level will go down, right? And then you go. So if you then look at one protein's expression level as a function of time, it'll go up and down and oscillate. So that is the idea. Okay. Does it make intuitive sense? So how do they build this? Well, they use 3 different repressors found in nature. One is called a log repressor, shown here as log I, and then they have PET R and then lambda CI. These are their names. Really, details do not really matter for this, but basically they do the same thing as I showed you before, except that now they have 3 components repressing each other. Okay. And then they have a reporter to show the level of one protein as a function of time, and they observe oscillation. Does that make sense? If you want, you can say that the protein 3 is linked to a GFP, okay? And that's your reporter. Now, actually, let me go back. But before they did the measurement, they actually wrote down a simple set of rate equations to relate the protein production and RNA production and degradation in a mathematical to each other in a mathematical form. These are coupled differential equations, and then you can use mathematics to predict what will happen, and for different sets of parameters. So you have a phase diagram and showing one parameter, in this case maximum proteins per cell, and the y-axis is the lifetime of the protein relative to the lifetime of the messenger RNA, and then based on these parameters, you can predict that only in this portion of the phase space you expect to see oscillations. Okay? If you look elsewhere, such as this point, then initial oscillation will quickly damp out to 0. So you don't expect to see oscillations here. So basically what it means is that if you want to see oscillation in your experiment, in their analysis, then they have to pick cellular parameters that match this phase space here. Okay? Now, as you can see, that is the ratio between the lifetime of the protein and lifetime of the mRNA. And that has to be small enough to be in this region of the phase space. Now, in E. coli, the lifetime of the mRNA can be just a few minutes, let's say 5 minutes long. The lifetime of the protein can be actually more than an hour. Okay? So if you don't do any engineering, then that number will be 20 or 30 or 40 times 40, and so it'll actually bring it out of this range. So what they had to do is to do some engineering to make the protein lifetime shorter than natural, okay, to make it happen. And to me, what was impressive about this was that they utilized a discovery reported in the literature just a few years earlier. So a group in MIT by Bob Sauer, they published that in E. coli, certain proteins are made with a tag, particular amino acid tag sequence. And if you have that tag, then protein degradation machinery in the cell can recognize that and then quickly degrade it. It's actually a quite interesting machine that has the engine here that is chopping up the peptide into small pieces. But then there's also ATP consuming motor protein here to pull the peptide into the channel for digestion, to unfold the protein and so on. But that's really topic for another talk. But the idea is that if you put that tag into the protein that you are expressing, the protein degradation become much faster. So the lifetime of the protein can become similar to the lifetime of the RNA in the cell. Anyways, after they have done all of these manipulations, and then yes, they could actually show that as a cell starts here and then divides to form a large colony, and then if you follow a single cell along its lineage, the fluorescence signal actually oscillates, okay, as a function of time. So that was actually quite an impressive achievement there. The reason why it speeds up over time is because still the degradation is not actually quick enough. Okay? It doesn't really fully degrade after one cycle is over, but seeing that alone is actually quite impressive. So that was published in year 2000, and then for 7-8 years, no one actually reproduced, or no one tried to do this in other systems. And one limitation was that oscillation actually damped up pretty quickly. So that was the best data they had, best typical data. And also oscillation was not really collective. You see maybe a few of them doing this a few cycles and then dying off. So the group in San Diego by Jeff Hasty, I think 08, they actually showed that you can actually make the circuits much more robust. So you can actually have all of them oscillate, making in phase and so on. So that was actually another really interesting development that happened much later. Questions?
Maybe I missed the part, but so when they tagged the amino acid tag to degrade the protein faster,
Yeah.
did they simply just genetically engineer some sequences in front?
Yeah. Yes. Yeah. Exactly. All right. So we discuss two classical papers from the year 2000, toggle switch and Repriscillator. And we'll take a break, and then we'll actually discuss another famous paper about gene regulation function that actually can give us some basis to actually go back to the toggle switch and do some mathematical analysis of that circuit. Oh yeah. Yes. So.
Can I ask you some questions about holiday junction?
Yes.
So, so I guess it doesn't matter the direction that you pull, actually, like these kind of directions, because the part you're pulling is going to align from the point that it's on, right?
Exactly. Yeah. Because the DNA molecule will rotate. to align the two, the vector connecting the two endpoints along the direction of the force.
Okay. And so when you pull this part, when you draw the energy landscape, do you think that it'll make it to come and stay at either of the heels so that it's easier to follow on to the next level? Is that why the K, two different Ks, they all increase as a function of force?
Oh, the third configuration. Yeah, I think that when you apply this way, it actually disrupts the stack conformations.
Is that what you're saying? Instead of having a square in the middle, you're going to have diamond? Like that?
Okay, that model came from relatively sophisticated structural modeling. You actually can parameterize the structure with two coordinates, both angular coordinates, and then based on the constraints from these three different experiments, you can actually model that. But yeah, this piece of data was critical in actually getting that model. Yeah.
Okay. So in terms of this energy landscape, the pulling is gonna, what exactly is it expected to do on the third point?
So I think what most likely what's happening when the pulling is to, to kind of shift them up like that, to destabilize the stack conformation.
Okay.
I think that's what's going on.
Okay. All right, thank you.
Okay, that's better. All right, so I'm going to talk about a paper from Michael Elovich. He's the one who did that Repriscillator paper as a postdoc, and then now he's independent in Caltech. So this is one of the really famous papers. So the repressor can affect the target protein's expression. In fact, it can reduce the target protein expression. Okay, so if you plot the concentration of the repressor protein, and then plot the rate of protein expression of the target protein, then you expect to see a certain function. Okay, so when repressor concentration is 0, okay, it'll have a certain value. So basically, that is the rate of protein production when DNA is naked, without any repressor bound to it. Right? When repressor concentration becomes infinite, then you'll have a complete repression, and then the rate of protein production will become approach 0, right? So it'll become very small phi here. So and then you'll see some curve like this. Okay, and this function is called GRF, gene regulation function. So So this paper was an attempt to actually measure this functional shape and parameters in vivo inside the living bacterial cell. Well, to do that, what you need to do is that you need to measure two values at the same time from a single cell. You need to measure the repressor concentration, and you need to measure the protein expression rate. And that you can do by fusing this guy to so you can fuse a repressor to a red fluorescence protein, for example, and then you can fuse a target protein to GFP. And then from single cell imaging, you just measure, the rate, these quantities, and then you can relate the two and plot one versus the other. Okay, so that is the idea. I think it's better to turn off the light. Okay, thank you. Okay, that is what you're after. Okay, and that is actually what you do. So this is the target protein that you're making fused to green fluorescence protein. It says CFP, but don't worry about that. It's green. Okay, and then the repressor that you're talking about is fused to red fluorescence protein. It says YP, but it's red. So just look at the color. Okay, and so these are two reporters for the repressor and then target protein expression. And what they do initially is that they initially make a lot of the repressor in the cell, so that the cells start with lots of red proteins. And you can do that by controlling the inducer amount and so on. But details do not really matter. But then as time goes on, and then you start producing the repressor. And then as time goes on, the cells will divide and divide. And what happens is that as the cells divide, first cycle of cell division, second, third, and so on, the level of the red protein per cell will drop, okay, by a factor of 2 more or less because of cell division. And then I told you that degradation of proteins in E. coli is really very slow. So that is not really significant enough to matter here. Basically, you do factor of 2 reduction in red signal as a function of time. But as the repressor level goes down, and then as the repressor concentration goes down, eventually it does not repress the protein production anymore. And eventually, this guy will be produced. Okay? And then later on, green will actually go up. So these are the snapshots of the data. You can see that initially they're all red, and then becomes less and less red, and then everything turns green. So that is the experiment. Does that, do you follow? Okay. Let me see if the movies work. So this type of movies are called time-lapse movies. And you have to be pretty good in taking good data, because unless you control the drift of the microscope stage and so on, these takes at least a day to do, and things will drift out of focus or drift out of the field of view. And you can also write your own scripts to identify cell boundaries and quantify the signals from individual cells. And here's another example. All right. So this means that you can actually measure these quantities quite well. In fact, they can do this for many, many different cells, and these are different cells giving rise to some different data points, but they all show very similar pattern. So you can get a qualitative result that matches your expectation, but if you want to actually calculate the this gene regulation function, then you actually have to know the absolute concentration of the repressor proteins in the cell, and not just the relative brightness of the signal. Okay, and well, and what they do is quite ingenious. If you have, a lot of proteins, let's say 1,000 proteins in a cell, and when the cell divides into two cells, then division of that 1,000 proteins in a random manner will still give you more or less half and half, 500 to 500. So the partitioning noise will be quite small relative sense. But if you only have 10 proteins in a cell, then when you divide 10 proteins randomly between two cells, then you can have a large partitioning noise. You can easily get 1 versus 9 proteins and so on. So by looking at the noise in partitioning, they can actually deduce how many proteins are there to begin with. So they, using that trick, they were actually able to build, calculate the apparent repressor concentration, and then they can actually use a mathematical function to fit the data and to obtain the parameters. So data actually are shown for 2 different conditions, and the function that they use is shown here, and I'll explain how you obtain that functional form in the remaining time of today. But the bottom line is that they could actually obtain all of the parameters from the data based on the living cell experiment. So this was the first actual demonstration you can get the gene regulation function from in vivo data. And I want to draw your attention to this one parameter called n, and this n is called the Here's coefficient, and n was more or less 2. Okay, that is consistent with the idea that 2 repressors can bind cooperatively to cause the repression. I know that these things can be quite confusing, especially to physics students. So, just let me know if there's anything. I can just go slow. Okay, can you have the light on? So what I want to do next is to kind of derive or rationalize why you have this functional form. Okay? And the R there is the repressor concentration. Okay? So let's say that you have n proteins of repressor binding to the DNA cooperatively to repress the gene expression. And I'm going to use D to denote the DNA, and P to denote the protein, the repressor protein. So then the reaction that we are talking about is this. And protein, DNA, binds to N proteins to form this complex, one DNA and N proteins. Okay? And then this binding can occur at the rate of K on with the appropriate dimension. And then this can also dissociate with the rate of K off. Now, we use a square bracket in kinetics to denote the concentration of whatever you're trying to study. Okay? So that will be the DNA concentration. Okay? So this denotes the concentration. So this is what you can write. Okay. How quickly is the concentration of this complex, N proteins bound to DNA complex as a function of time? Well, that will decay with this rate. Times the concentration of that complex. But it will also assemble with the K1 times the DNA concentration and the protein concentration to the nth power. In steady state, concentration of different components do not change. So therefore, but DNA found as part of that complex and DNA free is by itself, if you add them together, then that will be the total DNA concentration, total amount of DNA you have. So you can replace this by that. So if you do that, then total DNA concentration here. And by rearranging, So then the concentration of free DNA without the repressor bound is given by this expression. Notice that P is the protein concentration or repressor concentration. Now it's looking very similar to that expression there, because R is the repressor concentration to the nth power, and then everything else is constant. Okay, but why do we talk about this? Well, for the RNA polymerase to bind to start gene expression, DNA has to be naked, not bound by the repressor. Okay, so concentration of that free DNA is actually directly proportional to the protein expression rate. Yeah, because only when the DNA is empty, naked, without the repressor bound to it, then RNA polymerase can bind and make RNA, and then followed by protein production. So your production rate that you're measuring, gene regulation function, should be proportional to that free DNA concentration. So it's proportional to some So that's basically, right, I'm taking it from here. So that is a functional form that people use and that they use to fit the data. Okay? And the key point is that, what is that n value? And they got actually n equal 2, more or less. Okay? That means that somehow 2 repressor proteins have to bind simultaneously to cause the repression. And I think it's very likely that you can rationalize the finding of n equal 2. By going all the way back to the second slide here. That to get the maximum repression, you have to have the repressor binding to 2 sides of the unit simultaneously. And that requires actually 2 units of the log repressor that can bind together. The reality is a bit more complicated. It doesn't mean that you have a log repressor dimer doing this. In fact, what I'm showing you is a tetramer of the log repressor. And dimer can bind to 1 operator. And then when you have tetramer, then you can bind 2 operators. So in a sense, a dimer can be considered to be a single unit, and then it can form a dimer or dimer, and then bind 2 sides simultaneously to obtain the maximum repression. Okay, so on Thursday, Jan Schemner will give a guest lecture on optical trap and optical tweezers and its applications, and then I'll continue the discussion in the genetic circuits and oscillators and networks and so on next Tuesday.